Good afternoon, everybody. Today, I'm going to stick my toes into Dr. Levine's area of expertise, uh, and it's going to be very specific. Today, we're going to discuss genetics, and what I've tried to do is simplify things a bit so that you'll see later in the lecture the array of genes that have been linked to IBD. But we're really going to discuss the major mutations that we see and how they affect our response uh, essentially to bacteria, and how these mutations contribute to the clinical expression of disease.
I apologize for interrupting, but do we not have radiology rounds?
Sorry, is that not two? I thought it was scheduled for two.
I thought it
Usually
was after the.
one because we have pathology at 2.
You know what? You're right.
You have radiology now?
Let's ask Dr. Ruckin if we can
Yeah.
just reschedule for next week, and I apologize.
Do you
Okay.
want me to help? You want me to give this next week and you go to
No,
radiology?
Steve, why don't you continue and we'll just reschedule Dr. Ruckin for next week.
Okay.
We only have 3 cases in radiology, as I
Yeah,
understand
we'll
it,
just
too.
reschedule with Dr. Ruckin, and I'll send an email apologizing.
Okay.
So anyway, you've heard all of this before from Dr. Levine's lectures, but what is IBD? It's a chronic GI inflammation of unclear etiology, although we now know more about how it develops. The current working hypothesis is that it represents a dysregulated mucosal immune response I use the word commensal with some trepidation. It's actually resident flora. Commensal would really refer more to normal flora, and we now know that the flora in patients with IBD is different. But most important, it's our response to the microbiome in a genetically susceptible individual. And some, but not all, of the patients who develop inflammatory bowel disease have clear inflammatory triggers. So I know Dr. Levine has shown you this slide before. We have the intersection in this Venn diagram of microbial antigens, the immune response, environmental triggers, and genetic susceptibility. And some combination of this soup, if you will, when put together, will result in the clinical expression of disease. The conceptual framework is shown here. And at the top, we have genetic factors, the response to antimicrobial peptides, and we'll learn which of the genes is responsible for doing that. Autophagy, we'll talk about autophagy, how we handle bacteria, and how chemokines and cytokines are elaborated. And then we have the environmental factors. What are the microorganisms resident in the bowel? How does diet contribute? Other infections? Is stress an important factor? The use of NSAIDs. Obviously, we know that NSAIDs can cause bowel ulceration. How might they contribute? So there's a whole raft of environmental factors that are clearly can influence, if not determine, the disease. What appears to be consistent is that something has to happen to the bowel where one has impaired barrier function, which leads to the increased translocation of bacteria and bacterial elements. With this translocation of microbial products, immune cells are activated and we develop a chronic inflammatory response. Now, this chronic inflammatory response in most individuals is normal because it represents a state of controlled inflammation to deal with whatever bacterial elements end up penetrating the mucosal barrier. So when we get a report from pathology stating that the bowel shows chronic inflammation, take that with a grain, maybe a ton of salt, because often this represents the state of chronic controlled inflammation without excessive immune cell activation. However, if this gets out of hand because of excess production of pro-inflammatory mediators, the failure of regulatory mechanisms through FOXP3 and T regulatory cells, and the activation of T helper, T killer cells, and macrophages, this can lead to an overexpression of the inflammatory response, leading to the clinical spectrum that we know as inflammatory bowel disease. What is key here is that we know that patients who develop IBD have a different microbial profile than people who don't develop IBD. And at the top, you can just see the difference, both the decrease and increase in various species of bacterial flora that are consistent in patients with IBD. But at the bottom, keep in mind that this is not the only influencing factor. Smoking, antibiotic use, breastfeeding, contraceptives, appendectomy, vitamin nutriture, the consumption of certain foods or beverages, all can have a differential effect on either being normal, developing Crohn's disease, or developing ulcerative colitis. Now, we've talked about the hygiene hypothesis in allergy. Well, there's also a hygiene hypothesis in IBD. Having pets in childhood, living on a farm, having a large family size, drinking unpasteurized milk seem to be inversely associated with the risk of Crohn's disease and ulcerative colitis. Why is that true? Well, it's obvious because all of these events, are associated with increased exposure to antigens and microbial elements. And sort of we become tolerant of them and we develop a state of controlled inflammation. So when later on in life something happens and you're exposed to one or more of bacterial elements, you've already been exposed to them and developed a tolerance. So again, shown at the bottom right, there have been over 200 IBD loci in the genome that have been discovered in association with inflammatory bowel disease. However, in most cases, they only modestly predict what is going to happen. So what do we know about genetics and IBD? We know that there's an 8 to 10-fold greater risk of IBD among relatives of ulcerative colitis and Crohn's disease patients. Genetic factors make a larger contribution in Crohn's disease. So, for example, the concordance of Crohn's disease in monozygotic twins is about 30% versus 3.5% in dizygotic. With respect to ulcerative colitis, that contributing factor reduces to about 15% in monozygotic versus 4% in dizygotic twins. However, it's clear that there is some genetic component here. We also know that something called the founder effect, and I'll show that on a later slide, is likely in IBD. There's marked increased prevalence in Ashkenazi Jews. There's a positive family history in 5 to up to 25% of patients. And siblings with Crohn's disease, the relative risk is 13 to 36-fold, with UC 7 to 17-fold. If both parents have inflammatory bowel disease, there's a greater than 30% risk to the child by age 28 years. So first and foremost, you have to understand that IBD is a complex genetic disorder. One or more genetic loci interact with a variety of environmental exposures. Inheritance is non-Mendelian. It's not one-to-one because you carry a gene, whether you're heterozygous or homozygous, in the vast majority of cases, does not determine your clinical status. So mutations clearly increase disease susceptibility. And this is true not only of IBD, but of other complex genetic disorders, such as diabetes, multiple sclerosis, autism, schizophrenia, and bipolar disorders. So these neuropsychiatric disorders, again, it's even more vague, but certainly with diabetes and multiple sclerosis, we know that there's specific genes involved, which are inherently susceptibility factors. So the majority of IBD patients, however, have no family history of IBD. And most of these patients, when one examines the genome, have relatively low penetrance genetic variations. There are no genes and variants that mediate familial IBD. I guess we could say IL-10, IL-10R, and we'll talk about that a bit later, may be the exception to the rule. But there is no precise functional allele or causative gene in the vast majority of patients. As I mentioned earlier, the founder effect applies to the Ashkenazic Jewish population, where a high frequency of a specific gene mutation in a population is founded by a small ancestral group. And this is where we think IBD originated. In Eastern Europe, among the Ashkenazic Jewish population, and then through the diaspora, it's gone all over the world. So let's now start on our journey in gene world. And so the genes since 2001 that have been associated with IBD are shown here, and I've highlighted those that we will talk about. Those include, and I put just for your fun, what all of these letters mean. I sometimes scratch my head and say, you know, why are they naming it this way? But there it is. So here we have the JAK-STAT complex, ATG16L, which is an autophagy gene, IL-23R and NOD2. You're all familiar with these. And IL-10 is put in red here because it's sort of it's an outlier. This is the gene that was first described as being predictive of the development of very early onset inflammatory bowel disease. And we'll have more to say about that later. But here is just some, these are not all of the genes that have been associated, but these are the primary ones. And again, only the ones that are highlighted will be the ones that we'll talk about today. So let's now add host genetic factors. In this case, we have NOD2, and we'll talk about its role in innate immunity, and ATG16L1, we'll talk about that in terms of autophagy. So we have to have genetic factors along with environmental factors, and these determine the microbiota composition. In the normal situation, you have, over to the left, you have a large diversity of bacteria, less viruses, and increased fungal population, or at least a variety of fungi. And this leads to, at the bottom, mucosal homeostasis, a state of controlled inflammation and immunologic tolerance. It is this loss of tolerance that is going to lead to the clinical expression of disease. Over to the right, when you have any one or more of these mutations in association with appropriate environmental factors, this leads to a reduction in bacterial diversity, an increase in viral diversity, and a decrease in fungal diversity. And as a result, you get a dysregulated immune system, clonal expansion of those cells that produce pro-inflammatory cytokines and chemokines, and you can develop, in this case, Crohn's disease, where you have an activated inflammatory cascade leading to mucosal ulceration. And we'll talk about all of these elements as the lecture goes on. So the first thing you have to understand is that the immune system is pretty complex, and it involves three very seminal and basic areas of activity: innate and adaptive immunity and autophagy. Innate immunity, which is represented by the NOD2 gene, is a non-specific defense mechanism. It occurs immediately or within hours of antigen presentation and is activated by the antigen chemical properties. In the case of NOD2, the activation is a bacterial element, the outstanding one being muramyl dipeptide. And then we have adaptive immunity. Adaptive immunity, IL-23R, and we talked about the JAK-STAT pathway. We will again when we discuss the new approach to inflammatory bowel disease with the drug tofacitinib. So this adaptive immunity is an antigen-specific immune response and generally occurs within days of antigen presentation. Finally, we have the fascinating area of autophagy. Autophagy defines an intracellular process whereby we envelop and degrade or recycle both self, such as aged mitochondria, for example, and non-self, such as bacterial elements. They are either expelled from the cell or recycled within the cell, depending upon which element is being phagocytosed. How these genes were discovered is a result of both genome-wide association and whole exome sequencing. In genome-wide association, an immunochip is created, which is designed to include a certain number of single nucleotide polymorphisms with small insertions and deletions based on where we predict the problem is going to be. So previously identified genome-wide association loci for 12 immune-mediated diseases have been conducted, IBD, celiac disease, and diabetes, where the genome is sampled extensively for altered signaling. And the problem with the genome-wide association, and I'm sure Dr. Levine can say more about this, is that the genetic variation of individuals of non-European origin is underrepresented. Hence the disparity between white and non-white populations with IBD. Whole exome sequencing is a technique for sequencing all of the expressed genes in a genome. This is known as the exome. Now, the exome comprises only a very small function, very small component of our genome. Most of the genes, we don't know what they do. They're not generally expressed. So in whole exome sequencing, you select a subset of DNA that encodes proteins. These are known as exons. And as I said, humans have 180,000 exons, but they only constitute 1% of the human genome. So only 1% of the human genome is actually expressed. And then we sequence the exomic DNA using sequencing technology to try to find the key mutations in the expressed gene. Whole exome sequencing was conducted as a large ENCODE project where an encyclopedia of DNA elements was put together, and a majority of single nucleotide polymorphisms were, however, discovered in the close of IBD genome-wide association, and the majority of these SNPs are not in the exome. So the majority of the single nucleotide polymorphisms don't actually present themselves as genes that are expressed in terms of function. So only a small number of low-frequency variants contribute to the spectrum of immune-mediated diseases. And in the case of IBD, a lot of this is through regulation or dysregulation of FOXP3 and T regulatory cells. So let's look here at how genes actually are expressed in disease. The frequency of the gene is inversely related to the size of its effect. So here on the y-axis, we have the magnitude of the effect of the mutation, and the allele frequency is shown on the x-axis. Not surprisingly, those gene mutations which are most common, and I'm looking at ATG16L1, remember this is autophagy, are clearly risk alleles, but do not determine disease. So you can have modifications in ATG16L, and that's not rare at all, but because it is so common, it really is not directly associated with disease. On the other hand, if you look at very rare mutations, and I'm looking at IL-10R, those individuals who have mutations in IL-10R have a very high likelihood of having a disease-associated genetic variation that is clinically relevant. So IL-10 is a rare mutation in the gene for IL-10R and IL-10, IL-10R being the receptor and IL-10 being the ligand, that's associated with severe forms of Crohn's disease in infants. The loss of IL-10, IL-10R function results in severe disease, and these infants do not have an apparent environmental trigger, unlike patients who develop IBD at a later age. So this is termed very early onset IBD. And this one group of patients, and we'll again talk about that a bit later, may be successfully treated with bone marrow transplantation. And what does IL-10 do? IL-10 inhibits TNF-alpha production in macrophages and requires the presence of STAT3 and JAK1 and two distinct regions of the IL-10 receptor intracellular domain. So IL-10 comes and actually blocks the pathway leading to the transcription of target genes, which are responsible for the elaboration of pro-inflammatory cytokines. Mutations in IL-10R allow this to go unchecked. And if you remember when we discussed tofacitinib, tofacitinib tries to do pharmacologically what IL-10 does. Blocks the JAK-STAT pathway, leading to the elaboration of pro-inflammatory cytokines. And here is the JAK-STAT signaling pathway, and I'll read it for you. Many cytokines involved in the pathogenesis of IBD signal via the JAK-STAT pathway. Cytokine signaling induces phosphorylation and dephosphorylation of these proteins. These STAT proteins migrate into the nucleus where they activate the transcription of inflammatory cytokines via NF-kappa B, which you've all heard about. We'll show that a little bit later. JAK inhibitors, like tofacitinib, are new therapeutic agents that are currently in clinical trials to block this pathway, leading to signal transduction and the production of excess amounts of pro-inflammatory mediators. All of this, of course, is mediated through FOXP3. FOXP3 increases in FOXP3 result in the elaboration of T regulatory cells, which downregulate the immune response and block the production of pro-inflammatory cytokines. And FOXP3 is activated by IL-10R. When there's a mutation in IL-10R, failure to activate FOXP3, which results in a failure to cause an elaboration of T regulatory cells, an increased amount of T effector or T killer cells, and the production of excess inflammatory cytokines. And what ends up happening, again, in these IL-10 or IL-10R mutated patients is that we have a variety of endoscopic findings. These patients generally have very severe expressions of inflammatory bowel disease at a very, very young age. And I might ask Dr. Levine to comment on that a bit later, but this is the very early onset IBD that you've been talking about. So again, let's go back to our looking at the effector size of our variants. We've talked about IL-10, very low frequency, very high clinical effect. Let's now talk about some of these other genes. So there have been over 100 IBD genes and loci identified, only NOD2, which in patients who are mutation homozygotes, and there's only 5% penetrance here, have a 20-fold risk. Only NOD2 has a meaningful contribution to Crohn's disease risk alone. All the rest contribute modestly to IBD risk. So NOD2 is the most important of the other genetic variants that we're discussing today. NOD2, otherwise known as the nucleotide binding olimerization domain containing protein 2, which is the same as CARD15. The ligand, remember I said muramyl dipeptide, that's what activates NOD2. It's part of the innate immune response, is derived from peptidoglycan common to both gram-positive and gram-negative bacteria. So it's not antigen specific. It's this protein. It's highly expressed in monocytes, and in Paneth cells. What do Paneth cells produce? Defensins and other antimicrobial elements. So appropriate NOD2 expression is crucial for proper colonization of the intestinal microbiota through activation of Paneth cell mechanisms. And we know that NOD2 deficient mice have an altered microbial population and an increased bacterial load in their stool and in their terminal ileum compared with wild-type mice. So this experimental model confirms the fact that NOD2 is an essential gene that helps regulate our microbiome. And of course, here we have our friends, the Paneth cells, as shown here on a photomicrograph on the right and a cartoon on the left. And all these granules are these defensins and other antimicrobial elements released into the lumen to control inflammation in the gut and control the microbial population. So the Paneth cell is the site of origin of crypt homeostasis and inflammation. And again, if we have host genetic fact under normal conditions to the left, you have a healthy gut with a normal microbiota. They have anti-inflammatory bacteria. They include commensal and anti-inflammatory bacteria, which produce pattern-associated elements, which are recognized, and the harmful bacteria are controlled. Our gut lives in a state of physiological inflammation, tolerance, and homeostasis. But when we have abnormalities in specific host genetic factors, such as NOD2 over to the right, combined with appropriate environmental factors, that leads to the development of a so-called pathologic gut with altered microbial elements and a dysbiosis, all right? And in this case, the elaboration of defensins obviously may not be normal or may not be sufficient to regulate the microbial composition, which can be mucolytic and end up penetrating and translocating, leading to damaged epithelial, increased bacterial adherence, and a state of chronic inflammation. So it's a delicate balance here, and it is determined not only by host genetic factors, but also by a host of environmental factors that may be involved. And of course, NOD2 is involved in pattern recognition. And when we have pattern recognition, we have controlled production through NF-kappa V of pro-inflammatory cytokines and chemokines, which can help regulate the bacterial population and keep our mucosa and submucosa in a state of controlled inflammation. It is when this process is dysregulated or overactivated that the clinical expression of disease can take place. So if we look at Crohn's disease-associated NOD2 mutations in individuals of European ancestry, there's a 2 to 4-fold increase in heterozygous carriage of one of the major risk alleles, and a 17-fold increase when one is homozygous or compound heterozygous for a NOD2 mutation. And the frequency in Americans of European ancestry is 20%. So 20% of individuals carry one or more NOD2 mutations. So again, mutation in NOD2 is not a determining factor, but it is a risk allele and a contributing factor. So when you have a NOD2 mutation, this may involve impaired clearance of bacteria, as shown on the previous slide, leading to deeper or transmural inflammation. However, the loss of function mutations alone are not sufficient to cause Crohn's disease. And there's been no association of NOD2 mutations in Crohn's disease among Asian or sub-Saharan African populations. This appears to be unique to European populations, and again, in and of itself is not sufficient to result in disease. Let's turn now to autophagy, which I think is one of the more fascinating elements of our immune system. Autophagy involves the degradation of damaged organelles and proteins and also clears pathogens. When it clears foreign elements, we call it xenophagy. It's required for immunity to multiple different types of bacteria. And the ATG16L1 gene encodes a protein component of this autophagy complex. And in one case, replacement of threonine for alanine in one position has been associated with Crohn's disease. And here is a depiction of what happens to autophagy. At the top, you have an aging mitochondria. You have an intracellular vesicle which surrounds it, forms an autophagosome, becomes an autolysosome, and you have degradation, and you can have recycling of the various elements of the mitochondria. Below are what happens when you have bacteria or another pathogen. Same process, only in this case, the autolysosome ends up expelling from the cell the bacterial element. But the processes are analogous in both cases. Now, what are the inducers of autophagy? Again, toll-like receptor stimulation, other things that may damage the mucosal barrier, such as starvation, the elaboration of pro-inflammatory cytokines. All of these results in an activation of the autophagy complexes. And as you can see over to the right, here's what happens to bacterial elements that are autophagosomed. They are eliminated from the cell. So what else do we have here in our genetic variation diagram? The IL-23 receptor. So this is again interesting because it goes both ways, both to increase and to decrease risk. Again, if we go back here, IL-23, relatively common, and in fact, mutations here can actually result in a protective effect. So the IL-23 single nucleotide polymorphism results in a substitution of arginine with glycine in the binding domain for JAK2 and has the potential to alter downstream signaling events and responses. So if it influences the JAK-STAT pathway, what can it do? Well, the glycine substitution present in 14% of healthy individuals, again of European ancestry, reduces the risk of Crohn's disease by threefold. And for UC, slightly less compared with the arginine. So you have a substitution of glycine for arginine, and this results in a reduced risk. So here's one gene where a mutation actually can reduce your risk of developing inflammatory bowel disease. I find this extraordinarily fascinating considering all of the pro-inflammatory genetic substitutions we've discussed already. So here again is sort of an overview of everything. We have the innate immune response, NOD2, autophagy, ATG16L, adaptive immunity, IL-23R, and the resultant JAK-STAT pathway. TL1A is a protein involved in pro-inflammatory response. We now have a monoclonal antibody that is directed against TL1A, and that's now in clinical trials in patients with IBD. And then, of course, we have the contribution of the defective barrier, which allows the translocation of bacteria, and then IL-10 signaling, which again, via the JAK-STAT pathway, mutations can result clearly in very early onset IBD, severe disease. So if we look at genes and inflammatory bowel disease, we talked about IL-10 and IL-10R and very early onset IBD, but there are a whole host of disorders where early onset IBD has been recognized. The most important for you to know about, of course, is glycogen storage disease type 1b and its expression. And you have to know that the treatment of this disease is significantly different from the treatment of Crohn's disease in non-GSD type 1b. And I'm going to allow you to look that up for yourself because you're going to have to know that for the boards. And here's another whole host of diseases associated with IBD. And the list goes on. And again, here's the FOXP3, IL-2 and IL-10 signaling defects. And remember, the main FOXP3 disorder is IPEX syndrome, which is X-linked immune dysregulation, polyendocrinopathy, and enteropathy. And the enteropathy is similar to Crohn's disease. And of course, IL-10 we've already discussed in very early onset IBD. So if we look at common genes in the age of onset of monogenic inflammatory bowel disease, related to the genes I've shown you on the past slides, of course, the one that's the most common is IL-10 receptor. This is the number of all of these cases. As a single monogenetic cause of disease is the most common. Indeed, because this is expressed clinically very early, if we look at the number of cases in terms of age, you can see the majority of cases are between 0 and a couple of months. And that, again, is the VEO-IBD. The line graph is the proportion of patients with extraintestinal manifestations. So it's very interesting, and again, Dr. Levine can comment on this, that those patients with very early onset IBD tend not to have the extraintestinal manifestations that those who present later in life do. And this, again, looks at the management of patients with monogenic inflammatory bowel disease. Again, mostly IL-10, IL-10R. At the top, panel A, we have 750 subjects. 27% underwent bowel surgery. 21% got an enterostomy. And 63% had a resection and enterostomy. If we go down to those, again, same 750, 23% were put on biologics. And this, I think, is the most important. A lot of these patients undergo surgery, but biologics in monogenic disease tends not to be effective, only effective in 25%. And this, again, of the 750 that were followed, a collaborative study, they had 23% underwent a stem cell transplantation, 24% failure, but 71% improved. So again, this is the one group of patients where hematopoietic stem cell transplantation may be indicated for therapy. Keep in mind that IBD has significant disease overlap with a whole host of diseases. Some of the genes here shown in red and their mutations are shown common to all of these diseases with IBD, again to show you that we're primarily talking about risk alleles, not disease-determining genetic mutations. If we look at IBD susceptibility loci, there are a wide variety that are unique to Crohn's, a variety unique to UC, and then a whole host that are common to both. So keep in mind that those things that are going to determine the development of disease involve those factors that influence intestinal flora, both genetic and non-genetic, how nutrients can modulate the immune response, and how the microflora can regulate the inflammatory response because of antigen exposure, immune maturation, and the development of a state of controlled inflammation and controlled controlling the microbiota and controlling those elements that penetrate the mucosal barrier. So what can we summarize from today's lecture? Well, we know that there are a wide variety of loci that are associated with IBD. 70% of these are shared with other immune-mediated inflammatory diseases, but only a small fraction of heritability of disease is explained by any single genetic loci. And the paradigm of personalized IBD care will only be achieved if we further understand genetics, how the interplay between genetics and the environment can affect the microbiota and affect the development of specific disease states. And importantly, I think you need to understand the unique aspects that are presented by very early onset IBD. Sorry about that. Children with very early onset IBD can present with a different phenotype and more severe disease, they do, than older children and adults. Advances in sequencing technology have allowed for identification of causal gene defects, primarily IL-10, IL-10R, in a subset of patients with VEO-IBD. Identification of these variants has radically changed the approach in these children, mainly through the use of stem cell transplantation and the development of drugs to specifically address the problem. And these provide an opportunity for precision medicine by targeting the pathway responsible for disease. In this case, our knowledge of the JAK-STAT signaling pathway. And it is critical to understand the underlying defect in children with very early onset IBD. And again, remember that conventional therapy may not only be unsuccessful, but in the cases of immunodeficiency can be inappropriate as well. So I hope I've given you sort of a background of genetic determinants of disease. It still is a subject which is often confusing to me, and I think to all of us, but we have to learn as we go. So that is it for today. And I will stop sharing my screen.
Thank you.
Thanks, Steve. That was incredibly thorough.
Well, I get nervous when I give this lecture because it's
Yeah, it's a tough topic. I get nervous when I give lectures like that too. I think it's probably not a bad idea for probably me to develop a very early onset IBD lecture as a complement to this, because you're right that the management is very different and there are a lot of targeted therapies more coming onto the market every day. So it's not a bad idea for us to
well, that's why I sent you the lecture, because I thought it might be useful to you.
Oh, absolutely, it is. So I might steal some stuff from you.
That's why I sent it to you. Steal away.
Thank you.